Chapter 10 is on taxonomy and classification. I'm always mystified why they happen to put this information in Chapter 10 rather than earlier in the book, because we've already been talking about the names of bacteria. I really think that this should almost be Chapter 2. So I like to include this in the beginning part of micro rather than where it actually falls in the book. So taxonomy is the science of classification. Why is it that we actually need to have this classification? Well, first off, we actually have to have a way of standardizing names. There is actually rules around naming things. So if they do find a new organism, they actually have to follow the rules for naming them. I told you about the bacteria that I characterized coming from the leaves of plants of a plant in the desert in Israel. And there were a couple of organisms that we actually thought might be new, which was kind of exciting. And I was kind of joking around with my advisor one day, and I said, oh my gosh, I would love to name these organisms if we find out that they're new. And I said, I would love to name one of them Lysibacter richialis. Richie is actually my maiden name. Well, he looked at me and he said, no, there's actual naming systems for this, so we could not do that. I was joking. He didn't really get my joke, but there really is a system for naming things. So when they do find new organisms, they actually have to go through that naming system. The other thing is that it helps us to kind of group things so that we know how related organisms are. It can also help us to get some information about their evolution and their history. So if you notice this little picture in the corner, it kind of looks like an upside down triangle. This is really the big idea of taxonomy and classification. So that in those top groups at the very top, the big part of that inverted triangle, members in that group are going to share some characteristics. As we get closer and closer to the point of that triangle, they're actually going to have more and more things in common. So if you take a look at the naming scheme, so it starts with domain and then goes down to kingdom, phylum, class, order, family, genus, and species. So this is that classification system. So when I was in 7th grade, which was a very long time ago, our science teacher taught us a way to remember. So it's a little mnemonic, and it still is something that stuck with me to this day. So I always take the first letter of each of those, and you know how I love letter associations. So I always remember King Philip came over for good spaghetti. I've heard lots of different mnemonics throughout the years. I often wonder what happens in biology classes with some of those mnemonics that are out there. But really, whatever works for you, it's got to be something that you relate to. And this is something that I learned in 7th grade that I've kind of carried with me. If you ask me anything about classification, that's automatically what's going through my head. King Philip came over for good spaghetti. But if you take a look at that classification, as we get closer and closer to the bottom of the triangle where that species is, that means that those organisms are going to have more things in common. So that means that organisms in the same family are going to have more things in common than organisms in the same class. I mentioned that naming system, and we have a two-name system. It's called binomial nomenclature. And Carolus Linnaeus is actually credited as being the father of taxonomy and this binomial nomenclature. He liked to Latinize things. And so our scientific name for organisms, we also call the Latin name because they all have a Latin derivative. Carolus Linnaeus is actually not his real name. He kind of Latinized his own name. Believe it or not, at one time, organisms actually had 5 names. It's hard enough to think about 2 names of bacteria, like Listeria monocytogenes. It's hard enough to remember that. Imagine if we had 5 names. So Carolus Linnaeus kind of simplified it. So this binomial nomenclature is 2 names. So every organism has 2 names, and those 2 names are the genus and species. And there's some rules as to how you have to represent the genus and species, the Latin or scientific name. So genus is always capitalized. Species is never capitalized. It's always lowercase. It's always written in italics. If you can't italicize it, then you need to underline it. So if you take a look at some examples of organisms, so Homo sapiens, think about what that is. That's actually us. That's humans. Sometimes we call things by their common name, like we do with Homo sapiens. Escherichia coli, this is a bacteria that we work with quite a bit in lab. What helps is that we can actually shorten the name. So we can go by the first letter of the genus and then spell out the species. So Escherichia coli becomes E. coli. Much easier to remember and say E. coli than to actually say the genus and species. So here is that classification. So you can see those upside down triangles. And this is looking at several organisms. And as we go down that classification, if you take a look at the first triangle, this is us, this is humans, Homo sapiens. So we're in the kingdom Animalia. So what that means is that we have some things in common with other animals. As we move closer to the tip of that triangle, we're going to have more and more things in common. So for example, we would have more things in common with other primates than we would with mammals, because that order is going to be closer to that tip of the triangle, the species, than the class. You can take a look at two other organisms. So the triangle next to it, you can see the dog, Canis familiaris, and then the wolf, Canis lupus. You can see that they basically have everything in common until they start to diverge to different species. And then over that far triangle, you can see a bacteria and how that is classified. So the big idea is that as we get closer and closer to the tip of that triangle, organisms are going to have more and more things in common. So within bacteria, we can actually kind of further subdivide our species and talk about strains. So within species, we can actually have strains. So we can have different strains of bacteria. What that means is that they have little genetic differences. So in the previous chapter, I talked about the biochemistry in identifying a bacteria. We could actually have two strains of E. coli, and we could actually run them through those systems and look at their biochemistry. They would both type out as E. coli, but they could actually be different strains where they might differ a little bit genetically. And we see this with E. coli. So for example, there's different types of E. coli. So we have a lot of E. coli in our gut. That's different than the E. coli that we might study in lab that has plasmids. And remember, plasmids are little extra DNA. It just contains nice to have DNA, not necessary genes for the bacteria. And then there's also the really dangerous E. coli that we often hear about with recalls, those really serious recalls, which is E. coli 0157H7. That's the one that causes that hemorrhagic inflammation of those really serious diseases with recalls. So within species, we can actually have strains. Strains of bacteria are kind of like breeds of dog. So we just looked at that Canis familiaris. So if you have dogs, they are going to type out as that particular species. Of course, we know that we can have different breeds of dogs. So you could have a poodle, you could have a cocker spaniel. We can also have some amalgamations as well. So you could have a cockapoo, which is sort of a hybrid of a cocker spaniel and a poodle. So strains within bacteria are kind of like breeds of dogs. So how is it that we actually define a species? So species within animals is a little different than bacteria. The way that species are defined in animals has to do with whether or not two individuals mate and they can actually produce offspring that can then go on to reproduce. So are they fertile offspring? So they would have to be able to go on to reproduce to be considered a species. So if you've ever seen some different organisms like a liger is a hybrid between a lion and a tiger. Now, a lion and a tiger are two different species. So it's not like we could have one liger mate with another liger and get more ligers. These are huge, immense animals. I once saw one at a fair. They are huge animals. So they would actually be different species, lion and tiger. So it's not like that lion and tiger, that liger could actually go on to mate with another liger and make more ligers. So that's typically how we define species with animals. So why isn't this definition going to work for bacteria? Well, if you remember, bacteria reproduce by way of binary fission. So there's not a lot of genetic diversity. They clone their DNA and split into two. So we're not combining genes from different sources. There's no mating. So that's why that definition isn't really going to work for bacteria. So we have to find some other ways to define the species and bacteria. And I know that we spend a lot of time looking at our bacteria underneath the microscope, but again, that biochemistry, the genetics, its structure, all of those things can help us define the species of a bacteria. We could even look at immunological features. So if there were certain antibodies that bound to antigens on a bacteria, that would also tell us if they're related. So it's a little different to identify species of bacteria than it would be for animals, for example. So this is the 5 kingdom system. So we have plants, fungi, animals, protista, those are protists like pond swimming things, and then Monera. So with our bacteria, we're really in the world of Monera. So what is actually in Monera? So first off, our bacteria that we are looking at in lab, they really are going to fall under our eubacteria or true bacteria. This is also where we would see those pathogens. So things like E. coli, Staph aureus, Salmonella, they would all be considered eubacteria. There are some cyanobacteria. If we were doing an environmental micro, we would probably talk a little bit more about these cyanobacteria. They're going to be photosynthetic autotrophs. An autotroph basically means it can make its own food. Auto is self and troph is feeding. So it can actually make its own food. We'll talk a little bit more about another group of bacteria, which are called archaebacteria. These are more ancient bacteria and extremophiles. Their cell wall and their enzymes, for example, the enzymes they would use for transcription, are a little different than our eubacteria. And I'll talk a little bit more about those and the 3 groups. So here's our different bacteria. So here you can see we've got our gram-positive, gram-negative, and our spirochetes. And we're already seeing some of those examples in lab. Here's our kingdom protista. So if you've ever looked at pond water underneath the microscope, those little pond swimmy things more than likely fell under this category. We'll be talking about more protists when we get into the parasitology chapter. So things like Plasmodium that causes malaria, that is a protist. More to come on that. Fungi, we will also talk a little bit about fungi with the parasitology chapter. So there are some fungi that can cause disease, like Cryptococcus or aspergillosis. So we'll be talking a little bit about those. Plants, yes, we don't talk a lot about plants, but plants are important to life in general. Why do we really care about plants and microbiology? Well, because a lot of the drugs that we treat bacterial diseases with actually come from plants. And we talked a little bit about that with quinine coming from the bark of the cinchona tree. And of course, there's animals, probably the group that we are most familiar with. Many of the things that we talk about in microbiology, of course, are going to affect animals, particularly in our clinical micro class. We're mostly concerned about the effects on humans. So a little bit more on the archaebacteria or the extremophiles, the ancient bacteria. There's actually three types of archaebacteria. So there's methanogens. They're anaerobic, so they don't need oxygen. They can live without oxygen, and they actually make methane. So if you've ever driven by a landfill, you've probably seen those little pipes that are coming out of the landfill. That's actually so that methane can escape. So we would find methanogens in landfills, and what they can decompose, and then they can actually produce methane as a byproduct. When I took microbiology, I had to write a paper, and it wasn't a class where we could actually pick our own organism. We were actually assigned an organism. And so the organism that I was assigned was actually methanogen. I still remember to this day, the name was Methanobacterium thermoautotrophicum. And this is the type of organism that you would actually see in a landfill. I'm not going to make you write a paper on Methanobacterium. I'm not going to make you write a paper on any bacteria, but I did have to write a bacteria, and that's actually where it would reside. I also had some experience with some halophiles. So I told you about my bacteria from the desert. They were halophiles. They actually liked salt. So they tend to be aerobic bacteria, and they can live in high salt conditions. So they have a way to kind of take care of those situations where most organisms would actually be killed by that type of salty environment. So I talked a little bit about those halophiles that I studied from the desert. Then we've got our extremophiles, our extreme thermoacidophiles. They can actually tolerate really high temperatures. So these bacteria we might see in hot springs or hydrothermal vents. Now, in order for them to withstand those really high temperatures, they actually have to have extremozymes. They have to have enzymes that can actually withstand those high temperatures. So we call those extremozymes. We actually have a lab process that takes advantage of these extremozymes. That lab process is called PCR. You've probably heard of PCR with forensics. They do this like if you send out your DNA to like 23andMe to find out some information on your genetics. So PCR is short for polymerase chain reaction, and this actually uses an extremozyme from an organism. So in, the late 80s, early 90s, the scientist Kary Mullis was trying to develop this technique to replicate DNA, trying to replicate what the cell already does with DNA replication. So he was having a little difficulty with replicating the DNA because the procedure required heat, and the enzymes that he was trying to use were not tolerant of that heat. And we know what happens with enzymes when they get to really high temperatures, they actually denature. So Kary Mullis was kind of an eccentric scientist, and he was actually having an argument with his girlfriend. And for some reason, it occurred to him that, hey, maybe I can actually use these extremozymes from these acidophiles, these thermoacidophiles in the procedure. I often wonder what that argument was about that actually made him think about these thermophiles and these extremozymes. He ended up perfecting the technique, which has completely changed science in diagnosing diseases and forensics and things like 23andMe by developing PCR, and he won the Nobel Prize. He kind of threw some shade at his girlfriend when he was accepting the Nobel Prize and said if she wasn't such a you-know-what, he probably wouldn't have thought of that. But we actually can take advantage of these thermophiles in lab techniques like PCR. We also know that viruses cause disease, and we will be talking about viruses. Viruses are classified a little differently than living organisms like our bacteria or plants or animals. They are not living, so they don't really belong to any kingdom. Because of molecular techniques and knowing whether or not the virus is an RNA or DNA virus, that really helps us with classifying to find out if they are DNA viruses or RNA viruses. Viruses are very simple structures. They're not cells. They're a nucleic acid, either RNA or DNA, surrounded by a protein coat, which protects that nucleic acid. So since viruses are not living things, they are not classified the same way as living organisms. We're mostly concerned about classifying and talking about bacteria and microbiology, and studying the evolution of bacteria can be really difficult. Why is that? Well, first off, if you've noticed in lab, they actually look very similar. Cocci, a circle, looks like a circle. So they all look very similar. So their morphology is similar. They also reproduce very quickly. So we talked about that generation time, where bacteria can double their population in a matter of an hour. So that means that they can evolve very quickly. So it can be really difficult to track. And one way that scientists can study evolution and how related organisms are is through the fossil record. These are very tiny things, so there really isn't a lot of fossils. So really studying the evolution of prokaryotic cells can be really difficult. It can also be difficult to identify organisms, and I certainly know this firsthand in studying the bacteria from the desert and characterizing them. There are some different ways that we'll talk about where we can characterize and we can identify organisms and find out how related they are. So a very simple way to organize bacteria and find out what it is that you're actually looking at is through a dichotomous key. This is not really going to give us a genus and species name. It's going to give us sort of like a category. And this is using yes/no questions. So this is not like using open-ended questions. This is like, is it a circle or is it a rod? This is not, well, where does it grow and what type of food does it like? Not open-ended questions. If you've ever done any hiking and you've tried to identify plants, sometimes you can get a dichotomous key to identify plants, like does it have 3 leaves? Does it have 5 leaves? So this is going to give us sort of a general area. So if you take a look at the first one, it's asking, is it gram-positive? So if it's gram-positive, you're going to go to number 2. If it's not gram-positive, you're going to go to number 3. So by going through this series of asking like these yes/no questions, we can have an idea of what we're actually looking at. So whether or not we're looking at a gram-positive bacilli or whether or not we're looking at a gram-positive cocci. Numerical taxonomy is another way that we can find out if we have a new organism or if they're related, and this is by looking at patterns. So what we would do is we would assign a 1 and a 0. So if an organism has a particular characteristic, we would assign it a 1. If it does not have that characteristic, we would give it a 0. Now, it sounds very simple. Let's just look at the patterns of the 1s and 0s and find out how similar those patterns are. I can tell you from firsthand experience, it's a lot more complicated than that, and you really need lots and lots of characteristics. This is actually something that I tried with our bacteria. I did not have nearly as many characteristics. And you also need a computer to help you analyze the data. The only computer that we had was me to analyze those patterns. And it really did not give me any useful information. So this is useful if we have a lot of characteristics. We have a computer to analyze the patterns of those 1s and 0s, and that can tell us how related organisms are. If we had a greater match of like 90% of those patterns matching up, then that means that there's a good bet that those 2 organisms are the same species. Just from experience, I can tell you it sounds simple, but it's actually a lot more complicated than you think it is. Because of genetics and molecular biology, now that we know more about genetics, it makes it a little easier to identify bacteria. We could look at the base composition of organisms. We could also look at different DNA sequences, so we could probe for the uniqueness. The 2 that I'm going to spend a little more time talking about is DNA hybridization and protein profiles, or PAGE, polyacrylamide gel electrophoresis. So DNA hybridization looks at how similar the DNA strands are in two organisms. PAGE actually looks at the protein configuration. So separating proteins based on size, we can actually see a pattern. If that protein pattern is similar in two species or two organisms, then we would know that it may be the same species. So if we take a look at our DNA hybridization, so with this picture, if we see our DNA from two organisms, so our DNA is kind of this blue-green color from organism 1, it's kind of like this orange-red color from organism 2. If we were to actually separate those DNA strands and then we were to put them together like two puzzle pieces, if they matched perfectly, if we had 100% annealing, meaning that all of those bases are going to line up perfectly, then we know we have the same species. If we line them up and they mostly lined up, but some of those bases didn't really match and we had some bulging as we kind of matched up those two strands of DNA, then we know they're closely related but not the same species. If it's kind of like a hot mess and neither of those strands are really sticking anywhere, we don't have any matching, no annealing is happening, then that means that they are not related at all. With PAGE, we would actually look at the protein profiles. This is actually a technique that we do in Bio 109. We actually do this in a forensics lab where we're separating DNA out in different sizes. This actually takes proteins and separates them out. So the proteins would actually be loaded in a gel, and then they would move in the gel. So we would apply a buffer and an electrical field, and those proteins would actually go from one pole to the other pole. And based on the size, they would separate out and we would get a profile. So the larger proteins, they would actually move very slowly. So they would actually be towards the top of the gel. And the smaller proteins would move very quickly through the gel and they would be towards the bottom. So we would get this protein profile. If we were testing two organisms and looking at their proteins, if they have similar or same protein profiles, so we would actually see that pattern, and you can see the picture on the bottom, how those proteins would separate out. If those patterns are the same, it's a very good bet that it's the same organism. If they're completely different, then that means that they aren't related. There are also some ways that we can determine whether or not organisms are related. Let me just go back to this slide. We can look at ribosomes. So ribosomes are where proteins are made, and ribosomes evolve very slowly. The reason for that is that if ribosomes evolve so quickly, organisms wouldn't be able to get their proteins made. So we could actually look at the ribosomal RNA of organisms and find out how related they are. And we can actually look at 16S ribosomal RNA. So that's part of the structure of a ribosome. Ribosomes are made of different ribosomal RNA and proteins. So we could look at one ribosomal RNA, and that can tell us whether or not we have a new organism or whether or not we've identified it as a particular organism. So we actually did this with our organisms from the desert. We didn't do this testing in our own lab, so we actually sent it to a lab in Germany, and they tested the 16S ribosomal RNA, and they actually told us our identification. So that was a more definitive way of identifying our organisms. We could also look at immune reactions. So if there are particular antibodies that stick and bind to certain proteins, antigens on bacteria, if we have same antibodies binding to the same antigens, it's a good bet that they're related. We can also look at the different types of bacteriophages that bind. So bacteriophages, these are viruses that will attack bacteria. And in order for that bacteriophage to replicate, it actually has to attach to that bacteria, which means that that bacteria has to have specific attachment sites for that virus. So if we have bacteria where the same phage actually attaches, then that's a good bet that they are going to be similar. So thanks to a lot of molecular techniques and genetics, it's giving us a more definitive way of identifying bacteria.